Functionally Redundant Genes by Marissa, Anjali, Lakshita, and Jamisha. Before we begin, functionally redundant genes are defined as two genes that serve the same function in a single organism. There are conflicting theories regarding the purpose of these genes. Are they actually redundant, or are both genes necessary in order for the organism to be functional? There have been a number of studies conducted in order to determine the purpose of functionally redundant genes. Some researchers believe that the removal of one of the genes would not lead to a change in the organism. On the other hand, however, there have been studies that suggest that each of the redundant genes is completely necessary for the function of the organism. Removing one gene would create a behavioral or phenotypic change in the organism.
Two studies were performed on mice at the University of Utah in 2006. In the first study, the geneticists swapped the coding regions of the HOXA1 and HOXB1 genes in mice, which control brainstem development. This resulted in no visible change, which came as a surprise because not having either of those two HOX genes results in serious defects, like impaired breathing and losing nerves. However, this study was done without taking into account the behavior of the mice. On the other hand, the second study done with mouse barns revealed that there is actually a change in fitness that occurs when the genes are swapped. The mouse barns were set up in a way to force the mice to compete for space and food, much like they would in non-laboratorial conditions. Half of the mice in this experiment had two normal HOXB1 genes, and then the other half of the mice had their HOXB1 genes swapped with HOXA1 genes. The results showed that mice that had the swapped HOXA1 gene held 44.7% of mouse barn territories, while the mice with the normal HOXB1 genes held 55.3%. The reproductive ability of the mice with the swapped genes were also impaired. They only produced 64% as many offspring as those with the normal HOXB1 gene. This disproved their previous belief that the HOXA1 and HOXB1 genes had a redundant function and had no effect on the mice. In actuality, these two genes function in different ways. Swapping of HOXB1 genes with HOXA1 genes led to a change in fitness for one half of the mice. This destroys the notion that these two genes are redundant and can function as a backup of one another. Even though the mice with the swapped HOXA1 genes were still able to reproduce and hold territories, it was significantly less than the mice with two copies of the normal HOXB1 gene. This shows that there can be a change in an organism when two functionally redundant genes are swapped that is not necessarily phenotypically evident.
There was another study conducted in 2005 that turned to plants to explain functionally redundant genes. The pin family of proteins in plants contributes to the maintenance of auxin gradients throughout the plant. Because of its large role in the plant, the protein is present in many overlapping developmental processes. The gene for the pin family proteins are each recorded in different parts of the genome and have been thought to be functionally redundant. In order to test this theory, the genetic codes for several different combinations of the protein were removed or mutated, and the results were recorded. For example, one sample was grown without the pin 1, 3, 4, 7 gene codes. It was found that in the plant with every combination of pin gene, the expression of protein and the structure of the resulting plant was different. As an example, the organism where pin 1, 3, 4, 7 gene codes were mutated were more severely affected. This and several more comparisons of growth in plants with different genes expressed led to the conclusion that the genes were not functionally redundant. Although the resulting plants were all different, they were able to overcome the lack of pin protein and survive. However, it was found that the survival rate of these plants were not as high as those with all of the pin family genes.
Through both of these examples, we have concluded that functionally redundant genes are in fact not redundant. Without the presence of one of the redundant genes, the organism could face changes, whether major or minor. These changes do not necessarily result in a detrimental effect on the organism, but a difference is noticed. For the experiment regarding mice, the geneticists noticed a change in fitness levels among the mice, proving each Hox gene to have a different purpose. Similarly, in the experiment regarding the plants, every combination of PIN genes resulted in a different growth pattern. Each gene that was taken out to form these different combinations had its own function in contribution to the growth of the plant. Both of these examples resulted in a change to the organism. It is important to note, however, that the resulting organisms from the mutations were not as strong as the study with mice and had a lower survival rate regarding the study of the pin family. Quantitatively, this led us to the conclusion that if a functionally redundant gene was removed, a negative change would result. Furthermore, this suggests that because of the negative change, organisms with more genetically similar genes rather than redundant genes are not genetically favored or naturally selected for. This could account for why there are so many similar genes that are theorized to be functionally redundant.
And that concludes the end of our presentation. Thank you for watching.